We can hear you. So you are set to go. Recording is in process. Go right ahead.
Perfect. Thank you. So, hello, my name is Yuriy Sandmeier, and I will be talking about how RV3579C, a gene of Mycobacterium tuberculosis, or MTB, allows for tuberculosis to be inherently resistant to very common antibiotics like clarithromycin, known as CLR. The airborne pathogen MTB claims 1.8 million lives each year. In fact, prior to the current COVID-19 pandemic, tuberculosis, or TB infection, was the leading cause of death due to infectious diseases worldwide. Treatment of TB is difficult due to the need for multiple antibiotics over a 6-month period, which can lead to drug resistance and poor survival rates. Additionally, a major problem with treating MTB is its innate resistance to macrolides, a family of antibiotics including CLR that have excellent anti-inflammatory properties and are well tolerated in most patients. RV3579C may uncover why MTB has this innate resistance, and inhibiting this gene may increase the susceptibility of MTB to drug treatments, which would help us save millions of lives. To interrogate the genes of MTB, my lab developed the CRISPR interference system that represses target gene transcription. This system works by co-expressing a nuclease SG pyogene's Cas9 protein, or dCAS-SPY, with a single guide RNA, or sgRNA. Targeting specificity is determined both by Watson-Crick base pairing of the sgRNA and target DNA, as well as a short DNA motif known as the protospacer adjacent motif, or PAM, within the target DNA sequence. In the next couple of slides, I will show you how I employed this novel system to interrogate my gene of interest. Prior to my arrival in the lab, a genome-wide CRISPR interference screen was conducted, which identified RV3579C, shown here in green, that had not previously been described or studied in literature. Unlike most other candidates identified in the screen, RV3579C was found to be essential for growth in MTB. Therefore, it was postulated that it may serve as a promising target, since inhibition of RV3579C not only stop the growth of MTB, but may sensitize MTB to macrolide antibiotics. As such, I decided to analyze this gene. Since there was no known function for the gene product, I used the basic local alignment search tool, or BLAST, to identify any notable motifs or domains that would shed light on this protein's function. I found that RV3579C in MTB and RLMB in E. coli both contain what's called a SAM binding site. SAMs are commonly used by methyltransferases as substrates to provide a donating methyl group, and the presence of a SAM binding site is consistent with the predicted methyltransferase activity of RV3579C. In E. coli, this motif causes RLMB to serve as an RNA methyltransferase, leading me to suspect that both MS6073 and RV3579C serve a similar function in MTB. Extending this bioinformatics work, I used the software program AlphaFold to develop three-dimensional models for these three homologous proteins in MTB, with this protein being RV3579C, M. smegmatis, with this protein being MS6073, and E. coli, with its protein being RLMB. And this highlighted very similar structures, suggesting that both uncharacterized proteins in M. smegmatis and MTB likely do indeed serve as RNA methyltransferases. Given that MTB needs to be handled in a BSL-3 facility, and that miners such as myself cannot handle such agents, I tested my hypothesis using M. smegmatis, which is a well-characterized, commonly used model organism for MTB that can be handled under BSL-1 conditions. To determine if this protein was necessary for viability, I cloned 4 separate CRISPR-I sgRNAs targeting the M. smegmatis homolog, and demonstrated with a quantitative liquid growth assay that all CRISPR-I mutants showed severely impaired growth relative to the control strain, indicating this protein is critical for survival. Furthermore, there was a fourfold lower minimum inhibitory concentration, or MIC, for these mutants when exposed to CLR compared to the control antibiotic rifampicin, suggesting that the loss of this predicted RNA methyltransferase increases the susceptibility of MTB to CLR. In addition to the SAM binding sites, my earlier homology-based investigation and literature searches identified 5 residues that were highly conserved in the RNB family of N-methyltransferases, as well as in MS6073 and RV3579C. Using MS6073 and M. sphygmatis as a model for RV3579C in MTB, we individually mutated 4 sites from the listed residues noted in the figure to alanine, an amino acid with a simpler, uncharged, less bulky structure that does not contain any major functional groups. As shown here, no growth defects were observed after 3 days of growth, demonstrating that these conserved residues do not have any significant impact on the growth of M. sphygmatis, which suggests that these residues in its homologue RV3579C likely would not impact the growth of MTB. Next, I wanted to establish MICs for not only chlorothromycin, but for other ribosome-targeting antibiotics. In total, I assessed 14 different drugs, shown here, and I discovered that knockdown of MS6073 and M. sphygmatis lowered the MIC for fusidic acid as well as for the previously known CLR, suggesting that increased drug susceptibility is specific to certain ribosome-targeting antibiotics. Lastly, in revisiting the survivability curves which showed decreased growth when MS6073 was knocked down, I questioned whether knockdown merely prevented growth or actually killed the bacterial cells. To distinguish this critical difference, I induced knockdown and counted colonies at different time points for control, a knockdown, and a combination of knockdown plus a rescue construct, and found that although all 3 grew, the knockdown strain grew at a significantly slower rate. Therefore, the knockdown of MS6073 is bacteriostatic and not bactericidal. To summarize, first, using M. sphygmatis, we confirmed the essentiality of this gene for mycobacterial growth. Second, using structural comparisons, we showed that RV3579C and MS6073 are closely related to the E. coli 23S rRNA methyltransferase RLNB. Third, using individual mutants, we also confirmed the inhibition of MS673 renders M. sphygmatis sensitive to chlorothromycin. Furthermore, we also showed that various conserved residues between RV3579C, MS6073, and RLNB are not essential for growth of M. sphygmatis. Additionally, we constructed several MIC assays with ribosome-targeting antibiotics. In addition to the already known chlorothromycin, we also identified fusidic acid as an antibiotic that could potentially work alongside the chemical inhibition of MS6073. These data collectively indicate that neither knockdown nor CLR treatment alone will effectively treat these infections. However, a combination therapy of the two will work. Furthermore, using this model, and given that MS673 is structurally similar to the MTB homologue, it is highly likely that RV3579C functions the same way in MTB. As such, we believe we identified a novel method of combating TB infection by inhibiting RV3579C in the presence of chlorothromycin. With this new approach, we will increase the susceptibility of MTB to drug treatments, thereby inhibiting its growth and virulence and saving countless of lives. Here's a list of references that I've used when conducting this research. Lastly, I'd like to thank Dr. Victoria Shulman, Dr. Jeremy Rock, and my mentor Nick Poulton. And I would also like to acknowledge my birth parents to whom I would like to dedicate this work to. Thank you all for listening.
Yuriy, thank you very much for your presentation.